Welcome back, ladies and gentlemen. So, while the major discussion here, this chapter, has been about transcription and translation and the production of those proteins, we can't be remiss to forget that in order for those proteins to actually do their job, transcription and translation aren't the end all be all, right? So, translation can go through and use the genetic code, use that messenger RNA code, and produce us a polypeptide chain, but that functional protein doesn't have any sort of shape at that point. And so that polypeptide chain has to be folded and put into that three-dimensional configuration in order for it to actually do the job that it's meant to do in the cells. And it may mean that that polypeptide chain that is actually produced after translation in and of itself isn't the end. There are some additional situations where post-translational modification to that polypeptide chain still happens, and we need to be able to complete those proteins by whatever means that might be. So additional enzymes might cleave certain ends. We may need to bring those polypeptide chains together with other polypeptide chains. Really, this is coming back to that idea of protein folding. So this primary structure is what that messenger RNA is designed to create. But we then have to go through that secondary, tertiary, and in some cases quaternary folding to actually get the three-dimensional shape, those coils and folds um, based on hydrogen bonding, hydrophobic and hydrophilic interactions, sulfohydryl bonds, whatever the case might be, in order to make those functional in the shape that actually can do something in the cells. We also need to be able to target those polypeptides to specific locations. And a lot of that actually comes down to where that amino acid chain gets built, right? So remember that ribosomes are your site of protein synthesis, but those ribosomes can really exist in two different locations in your cells. They can be free floating in your cytosol, or they can be attached to your ER, more specifically your rough ER. And so when we look at proteins that are produced on ribosomes that are free floating in your cytosol, mostly those are going to produce proteins that are functional and stay within the cytoplasm of that cell. If they are made on ribosomes that are attached to your rough ER, then it's more likely that those proteins will go through that endomembrane system, right? ER to vesicle to Golgi to vesicle, and then get secreted from the cell via the cell membrane, or they might get packaged into lysosomes and remain within the cell itself. Cool thing about the ribosomes is that they are identical regardless of where we find them. So the ribosomes that are in your cytoplasm, the ribosomes that are attached to your rough ER are exactly the same, and so we can move them around, we can switch them from being free floating to bound, whatever the case might be, as the needs of that cell dictate. So because the polypeptide synthesis is almost always going to end up starting in the cytosol, the question is then where does it get finished? And unless those polypeptides contain signals to those ribosomes to attach themselves to the ER, then they'll finish their synthesis in the cytoplasm and probably be proteins that remain in the cell. However, there are signal recognition particles, or SNRPs, that will recognize specific signal polypeptides that are associated with that messenger RNA code that will then actually bring free-floating ribosomes and bind them to these translocation complexes on the rough ER's membrane. And when that happens, then that is a way for cells to tag that this protein, although its production started in the cytoplasm, this protein isn't meant to stay here. And so those SNRPs will actually take those ribosomes, attach them to the ER, and then allow the finishing production of that polypeptide to happen within the ER membrane so that that protein can then be packaged into those vesicles, go to those Golgis, and then ultimately get secreted out of the cell.